So again, before we get to this, you've been thinking and contemplating the relationship between a distribution and a sampling distribution. And now I can add a third thing in there and just say the distribution of a sample. Because there's three aspects when we're talking about distributions and shapes. But one, what does it mean? What is a population distribution? How would you explain that? Yeah, so you could have a whole set of data from something. Right. And typically we talk about, we say it's our population data. So if we had all the data about, like Simpson University has all of your data on something in their database. Right? And then they could look at that data in some type of a way. So what's one data point that they probably know about you?
GPA.
Your high school GPA, they probably have that on file somewhere. Yes. And then if we looked at all of that data of all your GPAs, we would talk about the distribution of your data. And if we talked about the distribution of GPAs, we could talk about things like its spread. Ooh, that moved around a bunch. And when we talk about spread, what do we have mathematical symbols that we use? Nope. Spread.
Oh, the shape.
Shape is another word.
The terms, I can't remember. It's skewed. Is it skewed?
Skew would be under shape, so we could talk about shape as being skewed. That would be one way we could talk about it. But a data is going to have a shape. It's going to have center. It's going to have a spread. There might be outliers. So IQR is one way to measure spread. IQR is one way to measure spread. What's another way to measure spread? Q1 and Q3 would just be data points within it. When we take the difference between them, Q3 minus Q1, that's the IQR. So the interquartile range, and this is a good thing to just remember because this is going all the way back to the beginning of the class. When we talk about range, don't say from here to here. You would say the range is 2. Which means, if I'm talking about the IQR, 50% of the data is spread out by 2 GPA points, if we're talking GPA. All right. If we're talking about range, like the overall range, also a measure of spread. Range is maximum minus minimum. We could say the range of the GPAs is 4.1, which means all of the data is spread out no more than 4.1 from each other. So the maximum minus the minimum. We don't say the range is from here to here. That might be how you've thought about it in the past. In stats, it's a very specific term. It is a singular value as the range. This is a symbol that talks about spread. That symbol is sigma. And it is the population standard deviation. The interpretation of standard deviation has been the average distance away from the mean, which means we have the mean, and that's where my mu comes in. Yep. And then outliers, depending on how we're doing it, maybe it was the 1.5 plus or minus the IQR, above and below the Q3 and the Q1. So this is a distribution. It's about the whole data. Now from the distribution, then we're going to talk about a sampling distribution, but the in-between step, right? Because then we have a sampling distribution. But in between the distribution and the sampling distribution is just the sample. So from a distribution, we take a sample. And the sample can have spread. What's the spread of a sample? We could still do the IQR, Q1 minus Q3 minus Q1. We could still take the range of a sample, but we don't have sigma. Instead, we have this would be the audience participation portion of class. Standard deviation of a sample. S. Okay, we can still talk about things being skewed. I mean, other words here would be normal. We'll talk about them being possibly symmetric. Still can have those same things here when we're talking about a sample shape. If my sample appears skewed, then it probably came from a population that was skewed. If I do a sample and it appears to be normal, it probably came from a population that was normal. So a sample tends to follow the same shape as whatever population it came from. And then lastly, or not lastly, what's my center in a sample?
The mean.
Yes, it's the mean. I saw someone mouth it and then just say it out loud. Nope, they're not going to. X bar. I can read lips somewhat. All right, X bar. And then outliers, it's the same calculation. If we're doing a box plot, 1.5 IQRs above Q3 or below Q1, we've done that in the past. And then lastly here, we have a sampling distribution. Sampling distribution is different. So here would be the, again, audience participation portion. If I talked about a distribution, talked about a sample, what is a sampling distribution? So before we get too much into all these other particulars about a sampling distribution, what is a sampling distribution? So the sampling distribution, correct, is the distribution of something. If I do a lot of samples, and if I find the X bars of all of those samples and I plot them, that's a sampling distribution. I can do the same thing with standard deviations. If I do a whole lot of samples, and for every sample I find a standard deviation, I can plot them all in one plot, and that large set of data is what's called a sampling distribution. And the sampling distribution will follow the sample size, which will follow the population size to a point. So if my sample sizes are small, my sampling distribution will be very similar to the population distribution. If my sample sizes get larger, the sampling distribution will approximately be normal. We spent a day looking at an online app where we said, hey, if my original population data looked very, very, very left skewed, and I did samples of size 2, and for every sample I got an X bar and I plotted like 10,000 X bars, my sampling distribution was also left skewed. But once the sampling size increased, it takes off some of those extreme values, if you will, because the more data points you have, the less effect any one data point has on that set of data. And when I found out that once I got to a place where n was at least 30, the sampling distribution was normal. Very common mistakes I see and I read, and you got to really work to eliminate this from your vocabulary, is I'll read and see people write, since n is over 30, the distribution is normal. And that would be wrong because the distribution is my original population. You have no ability at all, ever, to affect the shape of a distribution. A distribution has its own shape based on the data you collect. You can't make a shape. You cannot create the shape of a population distribution. The population distribution chooses itself. All right? Just like you can't choose a wand, the wand chooses you. All right? However, you can influence the shape of a sampling distribution by increasing sample size. And then when we got to S, then we're doing T, we had like this trifurcated. There were 3 different ways to do it. If n was less than 15, I had to have, if n was less than 15, I had to have a sample that I sampled that was very, 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 very nearly normal, because that meant it came from a population that was normal. If I had one that was between 15 and 40, that's x, not x, n. If n is between 15 and 40, then I can assume the sampling distribution is normal or close enough to normal as long as there are no outliers. So out here, there's no outliers for this one. Nope. And then lastly, if n is more than 40, once n is more than 40, I will assume that the sampling distribution is normal, and it doesn't matter if there's outliers or not. So again, statistics has been, I said this early on, is almost like its own language of math. It's very different than what you've done in the past. And being comfortable with some of the words that we use. And lately, I've talked much about population distributions. We've looked at samples, and we've graphed them to see if they, like, was the sample that we took from a normal curve or not? Did the data follow a normal curve? And then lastly, talking about sampling distributions and the effect that this has on sampling distribution shape. This is something that you should be very comfortable going through and figuring out quickly. If it's taking you a long time, you need to find ways to streamline your process of thinking through it. If you have to make your own flow chart so it makes sense, do that. If you like the outline format that I provided on one of those sets of problems that we did, then work your way through that. But figure out how you need to go about doing that to get to the place of saying, oh, I have 2 samples that were not paired, so it's a 2-sample procedure for 2 means, and then I realize that I don't know sigma for either of them, which means if I can, I'm going to use the T procedure, but then I have to look at the 2 samples and determine, are those 2 samples approximately normal? And if it's a small sample, I'm going to graph that data on a normal probability plot, see that my normal probability plot is approximately linear, and therefore say, since the normal probability plot is approximately linear, it came from a population which was normal, which means the sampling distribution is normal. Since it's normal, I don't know sigma, they were independent samples that were taken randomly, I can use the T procedure for 2 means. The T procedure for 2 means. All right? And all of these have their own nuances to figure out, am I using the right one for the right scenario? And then this is where we've been lately. Goodness of fit, independence. Today is homogeneity and variance. Homogeneity. From the same root is homo, which means same. Genus, same type. All right. Homogeneous would mean they are of the same type. Even though they're different, we're going to say that they are from the same type or they follow the same characteristics. So tests for homogeneity are done from samples that come from 2 different populations. When we did test for independence, the last time we met, test for independence was we took data from a single population. We sampled people from that population, and then we looked at data about them to see if they're is an independent relationship between some characteristics of data from the same population. Homogeneity, we're testing from two different populations. But if you looked at the slide from the last time we did from test for independence, same formula, same degrees of freedom. So between two populations, we can test multiple characteristics to see if they are the same or homogeneous between them. One example here, M&M's, brand name, they actually sell bulk assorted chocolate candies that, believe it or not, are pretty much the same shape, size, and color. They just don't have the little stamp on them that says M&M's, because you can't. All right? Probably made at a much cheaper facility with a lot more red number 5 and possibly a little bit of lead tainted in there. Maybe not, I'm joking. But if I were doing a test for homogeneity, my null hypothesis would be the distribution of colors of plain M&Ms is the same as bulk bin, right? And so my chart would look something like this: M&M versus just generic. And it would be red, orange, yellow, brown, blue. I think those are the only colors, right?
Green.
Are there green? Are there not orange ones? Yellow? M&Ms? Green? We'll call it green. I don't really know. I just eat them.
Me too.
I don't pay attention to the actual colors of them. I will, however, sort them when I eat them, and I eat them by color, as any logical person should. And then you eat them from lowest number to highest number, because, again, that would be logic. And if you're really math geeky, I am looking to see if there's a pattern in the colors that I follow. And for right now, to me, the ultimate handful of M&Ms would follow the Fibonacci sequence, which means there would be one of one color, one of another color, then 2, 3, 5, and 8. If that would happen, I would stop, take a picture, mark that day in my calendar, because that would be the perfect, ideal handful of M&Ms. It has not yet happened. And trust me, I've eaten a lot of M&Ms in my life, like a ginormous amount.
So every time you're just looking for that?
Every time I separate them out by color, and I'm looking for this pattern. Yeah. I've had a 1, 2, 3, 5, but then the others weren't 8 and 1, so you're going to get some close approximations, but the Fibonacci sequence is an amazing thing. But back to this. Red, orange, so the distribution of colors, they're going to be the same. So then I would look at how these are and compare them one to the other to see in a chi-squared test if they are about the same. The alternative hypothesis is that the distribution of colors will be different. Just like test for independence, except test for independence was, up here we're going to say da-da-da, they are independent, and down here they are not independent. All right. Another way to think about a test for homogeneity also is it's like doing a double test for goodness of fit. But let's look at it as an example. Here's my example. Do male and female college students have the same distribution of living arrangements? We'll use a level of significance of 0.05, right? That's our arbitrary alpha equals 0.05. We're going to test our p-value against that. Suppose that 250 randomly selected male college students and 300 randomly selected female college students were asked about their living arrangements, dormitory, apartment, parents, other. And here's the data. If that's my data, my conditions are, in this case, I first notice that I am taking it from males or females, which means that's different populations. Which means if I'm going to do something, it would be the chi-squared test for homogeneity. But let's continue. Are all of my values at least 5 in the table? 72, 84, these are all at least 5? Yes. All right, and was it a random sample? Because it says yes. Therefore, use chi-squared test, that looks like an F, test for Homogeneity. Going to use the chi-squared test. My hypotheses, males and females will have same distribution of living, what do they call it? Living arrangements. My alternative hypothesis would be males and females will have different distribution of living arrangements. Chi-squared equals the sum of each observed minus the expected squared over expected. And those are the first three things: conditions, hypotheses, and my formula. So then, in order to use the chi-squared formula, which is, again, chi-squared equals expected minus observed squared over expected. Oh, I forgot my giant sum. Sum of, I'm going to take the adding all of those up. I need to find the expected value, which means I need to find the row total, the column total, and then the total total. Well, I know my total is going to be 550, or it should, because there were 300 of them and 250 of them. And if I just look at females, let's say for a second, and let's just look at this one. All right, so have your calculators out as we go through this. I'll go ahead and figure out this total because that's easy. 3, so that's 163 that were in the dormitory. How many are females? 20, 2, 10, 18, 27. Is that right? I'm trying to add in my head, which you shouldn't ever trust that. Is that right? Oh, excellent. So to find the expected value for that one cell, I would take the row total, 300, times the column total, which is 163, and divide by the overall total, which is 550. And someone has their calculator out and they're figuring that out for me. Say a little bit louder. 88.9 exactly. 0909. So 88.09. 0.909 or 09? Okay, so 88.909. So then, as I think about calculations, chi-squared equals, these should be the other way around. It doesn't really matter because I'm going to square it, whether it's observed minus expected, but 91 minus 88.909 squared divided by expected 88.909 plus, and then I'll get a number at the end. You don't have to do all of them, but my expectation for you is that you can show me one of these things that's being calculated. That I know that, oh, yep, that's where the expected comes from. I can show you how to do one of them, and then we're going to rely on technology. I had an intermediate one here. So, yeah, so we're going to keep going here with this. So the calculations, we actually did it on the last slide to find out what these numbers are. We actually did this one here. My picture, because I'm doing chi-squared, it's going to look like this. And then I'm going to go to online. So what I would like you to do right now, if you got your device with you, which many of you do, if you don't, sit next to someone who does, pull up StatBlue. You're going to need to add a couple rows and columns. I did it already, so add a couple columns here. Dorm, apartment, parents, other, male, female. plug the numbers in, and go ahead, get it to kick out a response, then I'll go to the next slide. Want to talk about this for a second, degrees of freedom. So degrees of freedom, when we had a single test for goodness of fit, for the GOF, goodness of fit, degrees of freedom was n minus 1. For independence, the degrees of freedom was equal to the number of rows minus 1 times the number of columns minus 1. For tests for homogeneity, you can actually use either of them. Now make sure you note of this. This n is not 550. It's not how many people were sampled. This is number of groups or number of columns. So if I would do this one, number of columns, 1, 2, 3, 4. So 4 minus 1, that's 3. Number of rows, there's 2 rows. 2 minus 1 is 1. 1 times 3 is 3. If I do this, I'm asking how many things am I comparing? Dorm, apartment, parent. There's like how many groups are there? Not looking at this, but how many characteristics am I comparing? I'm comparing 4. So 4 minus 1, it's also 3. All right. So when you do this, it kicks out this number. Degrees of freedom was 3, tells me my p-value, which is what's shaded down here in my picture. Oh, this picture does not come from Stat Blue. That picture, if you want that picture, comes from Applets. The Applets page that we've used for like a bunch of things. It's the CHISQ.html. That's where that one came from. Where the degrees of freedom was 3, plug in the 10.1287, and it spits out the same answer, 0.0175. And then down here, I can look and see this was the largest contributing value to the chi-square distribution, which means the number of males who are living with their parents is far less than what you would expect. If they were going to be the same, that's the one that's the biggest difference, which could warrant further study for some other reason. What's my decision? What's your decision on this? What should we decide? Reject the null for someone who whispered it. This is small, so my decision, so my p-value, 0.0175, that's 01, hard to read that, is less than 0.05, which was that original alpha value that I said, testing it at a 5% level of significance. Therefore, reject the null. My interpretation, there is significant evidence to show that the distribution of living arrangements is different between the males and females. There is significant evidence to show that the living difference between males and females are different, because this should have only happened 1.7% of the time if they were the same. Since that's very unusual, tells us they're probably not the same. Test for variance. Here for a second. Sigma is standard deviation. Sigma squared is variance. So when we're doing our test for variance, is this idea that instead of testing for are the distributions the same, I can also use chi-squared to find out, is my variance what I expect it to be? So a test for variance is testing to see, is the variance what I expect it to see? Let's go. Test, my hypothesis test will be sigma squared equals something. So let's take a read. With individual windows, various windows at a post office, If you've actually ever been inside the post office, there's the lines, right? You can do the same thing if you're thinking about the grocery store. Individual lines at a post office finds that the standard deviation for normally distributed waiting times for customers on Friday afternoons is 7.2. Well, that's really specific. Yes, it is, because you can't just say waiting times ever, because waiting times are going to differ depending on day of the week, time of day, all sorts of things. So we find that this happens to follow 7.2. The post office experiments with a single main waiting line. So that's right. So everybody's all in one line, and then as a window opens up, they jump over to the window to see if that will lower the variation among the waiting times. 7.2 squared, because it says the standard deviation was 7.2. We're going to test to see if it reduces 7.2 squared. Biggest thing you got to remember when testing variances is they're going to usually give you the standard deviation, and you have to remember that actually variance is sigma squared, not just sigma. So if the standard deviation of wait times is 7.2, the variance is 7.2 squared. So my conditions were, it's a random sample. That has to be true. We have to assume that the underlying population is normal, which it says up here that it follows a normal curve. So that way we can use chi-squared test for variance. And then there's my slightly modified formula. Test for variance, much smaller. N minus 1, S squared, sigma squared. How many people? Random sample of 25 customers. Oh, so no, it's 25 minus 1, which is degrees of freedom. Amazingly enough, it shows up in the equation. That's on purpose. It's all part of the plan. 3.5 squared over variance, which is 7.2 squared. So like this. Take a moment, grab your calculators, find that number for me. Those of you not using your calculators either have a lot of trust or are just really lazy. I guess there's probably some other variations that could be in there. See, I'm so skeptical, I just don't trust anybody, so I have to do my own calculations most of the time, except for right now, when I'm relying on you. Anybody going to verify that? Bobbleheads in the room. I got a thumbs up. Okay. So that's the formula. Those are my calculations. I'm going to draw a picture. A chi-squared with 24 degrees of freedom is actually going to look fairly normal. This is chi-squared. It's going to look getting awfully close to normal. There it is. If you don't know what it's going to look like, you can go to the applet, look up the chi-squared, put in 24 degrees of freedom, and it goes like this. Probably a good idea. Maybe do that now if you got your device with you. Go to the applets page, find the chi-squared, put in 24, and what do we say that x value was? 5.67. 5.671, and then pick which is the right thing for the drop-down menu. Your drop-down menu here will either give you greater than or less than. So you got to think back. What was my null hypothesis? What was my alternative hypothesis? Pick the right one and see what it spits out as an answer. Because it's the alternative was less than, we got to make sure that this is actually x less than x. So make sure you've selected the right one. Anybody have that number? It's really small. Point 0.00004, which is really small. It's like remarkably small. Always going back and referencing my null and alternative hypotheses. That's why we start with them early on to know how it's guiding us in that process. 5.671 less than 0.0004. It's like, it's so small that on my picture, I mean, you can see right there, that's not, so 5 should be somewhere right about here, because this is 10. So it's this area under the curve, like it's so small we can't even see where the 5 would be, because this is so remarkably small. So if we assume the null is true, if we would assume that the standard deviation, or sorry, the variance, if we assume the variance equals 7.2 squared, what we saw happen should happen 0.004% of the time, which is virtually impossible. Not impossible. In stats, there's very few things that are impossible. All right? But my conclusion is, since the p-value is less than 0.05, it's actually like a lot less, but it's less than 0.05, so we're going to reject the null, accept the alternative. The interpretation, since the likelihood of this happening by chance is only 0.004%, that means there is significant evidence to show that the variances will indeed decrease by using the one-line method on Friday afternoons. Since that's so small, it would actually make me want to take a look at adopting this for not just Friday afternoons, but maybe all afternoons. Run some data, collect some stuff, and see if the variance actually decreases there as well. Practice, practice, practice writing those interpretations. Because you can hear me say it all you want, but until you try it and do it, you don't quite know, am I getting all the words on there that I need to say? Share it with one of your study partners, like, hey, what did you write? How am I? It's like, yeah. Check with the student who always gets the A's in the class and check with them. Last resort, have me read it. It's like, is this enough information, too much information? Is it making sense? So then with something like this, if this were a designed experiment where I randomly selected some days for this to happen, randomization, replication, had control on what's going on, could we prove that going to one line reduces variability in times? Absolutely. So a hypothesis test is one thing that we've done and looked at where we're just collecting data. But ultimately, when research happens and you do a designed experiment of some kind, you're eventually going to have some type of a hypothesis test to find out, did what I studied make a difference in whatever I was trying to prove? The chi-squared is a very common type of test to do when people are trying to look at, hey, does this have an effect on, these various characteristics between men and women? These various characteristics for those who get the old medication, the new medication. These various characteristics between this subpopulation of the United States and this one. Way beyond medical, marketing. Well done marketing should have aspects of this going into place to see, is what we're doing actually influencing and driving better sales? Right? It can be done in so many different places. Unfortunately, a lot of people don't because it takes time and energy and money. But those that do tend to find great benefit because your results actually mean something with a designed experiment, where you can say, I can prove this. Not just there's a correlation. There's lots of relationships that exist out there. And you can show that the relationship is significant, right? But until you have a stimulus and we're measuring a response, we're not going to be able to say that, oh yeah, it's actually cause-effect. I'm going to offer one point of clarification on something I said the other day. I did it right into class 2. I think a lot of people weren't listening because they were packing up. But to increase power, I can increase n, my sample size. If I have more things I'm sampling, I have a better ability to detect differences, graders, less thans. The other thing I can do is decrease, and this seems counterintuitive, decrease alpha. So if I don't care about my alpha, so instead of 0.05, I'm like, well, I'll be good with 0.08. If I'm okay with the probability of a type 1 error being 0.08, which is the same thing as decreasing my level of significance. So instead of saying, hey, this is less than 0.05, I reject the null. So I do this and say, now it's actually 0.07. I would still reject the null. But if I'm wrong, that would be a type 1 error. If I'm okay with that, it'll actually also increase power. And the power is the ability of a test to make the right decision.